Well, good afternoon, everybody. Welcome to this President's Lecture Series. This has a special significance. It's the first of our series in the calendar year 2018, and with that, the first in our 75th anniversary year as an institution, and a terrific way to get that year off to a start in hearing from our colleague, Dr. Phil Scherer. Dr. Scherer is professor in the Department of Internal Medicine and Cell Biology and holds the Gifford O. Touchstone Jr. and Randolph G. Touchstone Distinguished Chair in Diabetes Research, and he's the director of the UT Southwestern Touchstone Center for Diabetes Research. And given all that, it is a special pleasure to welcome Gifford O. Touchstone Jr. himself, a great supporter of UT Southwestern, obviously behind the ability to bring together the talent that constitutes the Touchstone Center and enabling the work that we're going to hear about this afternoon. Dr. Scherer is recognized internationally for his work in diabetes. Much of his work over the past 20 years is the outcome of his discovery of adiponectin in 1995. A hormone produced by fat, and which I'm sure he will do justice this afternoon in sharing how that has led to more insights into diabetes and the biology of fat and all of its importance that flows from that, including its relationship to heart disease and cancer. It was foundational work for his laboratory, and he has since defined the variety of important roles that fat cells play in relationship to both health and disease. His work has garnered appropriately international recognition. He was the recipient in 2015 of the Banting Medal, the highest honor given by the American Diabetes Association, and also even more recently in 2017, the Novo Nordisk Foundation Diabetes Prize for Excellence. His start on his life and scientific journey was at a distance from here. He went to undergraduate studies at the University of Basel in his native Switzerland. After completing his Ph.D. degree, he did postdoctoral work with Harvey Lodish at MIT, and thereafter joined the faculty of Albert Einstein School of Medicine, where he worked for 10 years before we were fortunate enough to recruit him to UT Southwestern. Since he has arrived at UT Southwestern, he has driven a dynamic program of research, again, within the Touchstone Center, but that's not all, as I have learned. So still, after 10 years learning new interesting things about UT Southwestern and its faculty, he's also passionate about basketball and has been one of the inspirations for a standing Friday basketball game between cardiology and the Touchstone Center. I'm not sure which skill he's going to share this afternoon, but I'm happy to turn the podium over to Dr. Scherer for his presentation entitled The Secret Lives of Fat Cells, Implications for Diabetes, Cardiovascular Disease, and Cancer.
Dan, thank you very much for the kind words and the introduction, of course. There we go. I think now I'm online, right? Thanks you, Dr. Podolsky, for the words. Talks on home turf are always a little more challenging than on the road. Naturally, I was thinking, okay, if I do a wonderful job today, chances are that you guys are going to forget about this lecture by the end of the week. If I mess up and I make a fool of myself, you will remember forever on home turf. So in that spirit, I hope you will forget this lecture by the end of the week. So what I want to do today is bring a little closer one of the cells that we love to hate. We all have too many of them. We appreciate that. Short of a tumor cell, there's probably no more malign cell out there in our system than the fat cell. And we like to think of them mostly as having too many in excess, and that gives us actually an increased chance to eventually get cardiovascular disease, diabetes, kidney disease, and in fact, it even raises our chance of getting cancer. So in that sense, I want to actually put a little bit of a different perspective into the fat cell, much of which is actually based on sort of a misunderstanding of what they actually do. And what I want you to walk away with at the end of this lecture is really, we want to keep our fat cells happy, and we want to avoid either making them sad or even worse, angry, because there's nothing worse than an angry fat cell. And in that, we want to learn a little bit more about the physiology of these cells and what it actually takes to keep our fat cells happy in this context. Now, very simple, of course, our fat stores, our fat mass, are the net reflection of what's going into the system in terms of energy intake versus the energy expenditure. And in terms of energy intake, of course, we all embrace our high-fat, calorically dense foods. And on the energy expenditure side, on the one hand, we expend energy just by laying in bed, basically. But on top of that, we have things like basal physical activity. So we run around, that burns calories. And then in these harsh Dallas winters, of course, we have to keep warm. And through adaptive thermogenesis, we also actually burn some calories. And you'll see that turns out to be very important. Now, the net balance, of course, goes into the fat cell. And these fat cells, we expect them to have a certain level of metabolic flexibility, just like the whole system. And when we say metabolic flexibility, we really mean by that the ability to adapt to feeding and fasting, which are fundamentally different metabolic conditions. And the system really has to maintain a high level of that flexibility. And that is critically dependent on how well we retain the ability of our fat cells and the fat tissue at large to expand and contract under the different conditions. Now, conventionally, when you open a textbook, and that was certainly the going rate 20 years ago, what is the function of the fat cell? It was basically considered to be flubber. Its primary role in the fed state would be to absorb sugars and lipids and store them in the form of triglycerides in lipid droplets in the fat cell. On the other hand, in the fasted state, we expect the fat cell to respond to the low systemic insulin levels that are in the system, and they're supposed to release these lipids, feed the liver, which is going to convert them into glucose and sugars, which then in turn actually feed that sugar to the brain, which is critically dependent on the supply of sugar in that fasted state. But that has actually changed quite a bit over the last 20 years, and we've gotten to appreciate the fat cell as a very complex biochemical machine that can actually produce a whole host of different things beyond just releasing lipids that it has previously stored. So it can act as an endocrine cell. It will release hormones, and we refer to these hormones that are highly selectively produced in the fat cell as adipokines. It will also release lipids, but not lipids necessarily meant to be used for energy. They're actually signaling lipids that communicate with other tissues. And then we'll also briefly talk about other unexpected metabolites that fat cells will release into the system during times of need, when the liver, for instance, is busy doing other things. So in this process of expansion, as we gain weight and expand the fat tissue, one of the critical questions is really, why do some of us develop these secondary complications such as diabetes and cardiovascular disease and others don't? So what really goes wrong in our adipose tissue in the process of this expansion? And we've learned a fair amount and defined the critical steps for the expansion process, one of which is really the ability to appropriately supply both oxygen and nutrients to the expanding fat mass. So an expanding fat tissue is almost the equivalent of an expanding solid tumor mass. It experiences the same kinds of stress under those conditions. So the vascularization is very important, the contributions of having enough oxygen in the system, because if we don't have enough oxygen, the cell undergoes a process that we refer to as hypoxia. And that hypoxia has a lot of negative implications for the fat cell. And as long as we have enough oxygen there, we actually have a lovely interplay between the fat cell and a lot of other surrounding cells, including, for instance, these macrophages, which we can divide in a simple way, maybe overly simple way, into two categories. There are the good macrophages and the bad macrophages. So the M2 type macrophages that are involved in rebuilding the tissue would actually be the beneficial macrophages under those conditions. If that vascularization is not properly taking place, the tissue will experience hypoxia, and secondary to that, it starts to produce a lot of these extracellular building blocks that create fibrosis. And with that, you have an increased rate of cell death of the fat cells that will attract the wrong kind of macrophages, the M1 type macrophages. So depending on how well we do this expansion process, we may or may not experience that hypoxia. And as long as we can postpone that hypoxia, the insulin resistance that occurs eventually in an expanded fat mass can actually be postponed, whereas if you have early stage fibrosis, the insulin resistance will kick in much earlier and ultimately result in that much dreaded inflammation that starts out locally within the fat tissue and ultimately spills over systemically into the system. So functional versus dysfunctional fat differs in many ways. There's a lot of other stresses involved, and if you look at this here with the basics, so we refer to it as the triad, the ominous triad of tissue dysfunction that involves the vascularization, the inflammation, as well as the fibrosis. And you can see here at the bottom, there's a host of immune cells that are critically involved in that process of either maintaining overall health in the tissue or actually pushing it over the edge and creating a lot of dysfunctional adipose tissue. So when you look at this slide here, which is a scanning electron micrograph of adipose tissue, you appreciate that it's not just the fat cells, so it's the whole vascularization that I mentioned, many other cell types with the immune cells, and most importantly, this very dense network of fibers that you can see here surrounding the fat cells that are actually this fibrosis that I was mentioning. And the vital role, basically, of the modulation of this very tight extracellular matrix is very critical to maintain the proper function of adipose tissue. And you can see this, for instance, in a collaboration with my colleague Sam Klein at WashU. He has actually taken individuals and subjected them to a very stringent weight loss program. They were all obese to start out with, partially dysfunctional, and we stained here for an extracellular matrix for a fibrotic component. And as you can see, at baseline, every single fat cell is surrounded by that fibrosis. And as he puts them on a very stringent weight loss program, you can actually see 5% weight loss, 10% weight loss, eventually 15% weight loss within the same individual, results in basically resolving that fibrosis. And with that fibrosis as a correlation, you actually see fairly dramatic improvements in metabolism in this very same individual. So fibrosis in fat is as much of a pathological phenomenon as it is in the liver or in the kidney that are more conventional tissues that we think about in the context of fibrosis. Of course, the tissue as a whole is also quite complex. We all have heard that some of us are pear-shaped, others are apple-shaped, depending on where the major location of your fat expansion takes place. And we could argue that in the context of fat, the same basic rules apply as with real estate. It's basically location, location, location. And from that perspective, it can't be emphasized enough that absolute assessment of absolute fat mass is relatively meaningless. We really have to look closer, and what matters is not the quality, it's the quantity, it's the quality of the fat that really matters. So in this particular plot, you can see we have plotted insulin resistance versus fat mass. And where we all want to be is, of course, in the lean, healthy. We have limited amounts of fat. We are limited amounts of insulin resistance. Most of us, of course, as we progress through life, end up in this category here. We have too much fat, and we are metabolically unhealthy and obese. Nevertheless, some of us get lucky in that sense and can actually gain a significant amount of weight, yet we do not have any signs of diabetes or cardiovascular disease, and they are referred to as the metabolically healthy obese or fat and fit individuals. And depending on the age group, you have anywhere from 20, 25, 30% of all of us will fall into that category. Perhaps the most striking example of how much we need our fat really is, of course, if we don't have any fat. So in the form of lipodystrophic individuals that have very limited amounts of fat storage capacity, and those actually, despite having very low fat mass, display exorbitant levels of insulin resistance with a really fatty liver and all the other secondary sequelae to having a metabolic dysfunction. Now, those are all clinical phenomena, and of course, what we try to do in our center and in my lab specifically is to address these questions at the level of a preclinical model. So the mouse model is really our key tool because there we can actually ask specific questions regarding cause and effect relationships that we can then take back into the clinic and validate them as correlations. So we have lean and healthy mice, of course. We have obese mice by simply feeding them a high-fat diet that we expose ourselves to as well. We have lean lipodystrophic mice, genetically modified, such that these mice don't have any fat available, and they display the exact same phenotype that human lipodystrophics would display. And then, of course, this category here, fat and fit, we have mice that are fat and fit. And in fact, we hold the record here at UT Southwestern for the world's fattest mice, and on multiple occasions. So we have mice that fit exactly the category. They will be the mouse equivalent of an individual that is 500, 600, 700 pounds. But what makes these mice so nice, not they're just cuddly and lovely to hold and very and always in a good mood, but they also actually display a lot of the characteristics that we see clinically as well in fat and fit individuals. So when you subject them to this checklist here, you will actually see, you look at their fat tissue, and you will actually see that they have, instead of having fewer big and angry and inflamed cells, they have a lot more fat cells that are smaller. They have, as a result, a reduced level of inflammation. They have very limited fibrosis in their fat tissue, and as a result, of course, that fat tissue, despite being really, really big, is very insulin sensitive. And when the fat tissue is insulin sensitive, that will translate immediately into all the other tissues. So the liver is very happy. We don't have liver lipid accumulation. Refer to it as steatosis. There's limited steatosis. And of course, the insulin-producing beta cells are actually also very happy because they have to fight with a very limited resistance at the level of the systemic insulin sensitivity. So expanding fat is, in principle, a good thing because it will protect all the other organs from lipid accumulation, including the liver, where you really don't want to store excess lipids. So that is in principle a good thing, and it hints at the complexity of the tissue in that sense. But there is additional complexity involved with fat. We now know, and we certainly had known that in the mouse for decades, that there's more than one type of fat cell. So we all think of it as the white fat cell that we see up here as being the conventional fat cell. But of course, mice were known to have brown adipocytes, and unlike the white adipocyte, whose major role is the storage of energy, the brown adipocyte is supposed to keep the mice warm. It burns energy. It has a lot of these mitochondria in it. And then we also realized that there are additional types of fat cells that sort of are a hybrid between the white and the brown one. And because they're a hybrid, they have a fewer mitochondria, but they have the ability to generate heat as well. We refer to these as beige adipocytes. Now, this used to be thought of as a phenomenon primarily relevant for the mouse. We knew that human babies have brown adipose tissue as they're born, but we really traditionally thought of this as being irrelevant for the adult human being. However, the radiologists actually taught us differently, and in a wonderful collaboration with my colleague here, Orhan Haas in radiology, who routinely uses PET scans for diagnosis of tumor lesions, you can actually see in this representative individual that there is a lot of different areas within the body that light up when you feed this individual or infuse this individual with radioactive glucose in this particular case. And these are actually these notorious newly discovered, they've always been there, but not appreciated beige and brown fat tissues that humans display. So this idea that perhaps we can convert our white adipose tissue into grayish and brown adipose tissue is, of course, fundamentally attractive from a pharmacological perspective, because you could think of ways by which we can stimulate this process, and simply by sitting in the couch and activating our brown adipose tissue, we could randomly burn energy, basically, without necessarily having to go through an exercise. So this, in principle, is an attractive concept. Now, people, of course, have been trying at many levels, again, in the mouse and in humans, to find ways by which you can stimulate this beiging or browning of the white adipose tissue. And there's a host of different factors that we have identified. Many others have contributed additional factors that, at least in rodents, will actually very actively produce and convert white adipose tissue into this brown adipose tissue. And it's still an open question in that sense, whether or not this is going to be an attractive weight loss regimen for humans as well. People are certainly excited about it and are working on it. Minimally, we can say the conversion of the white adipocyte to a beige adipocyte has potent anti-diabetic effects that it brings with it. Now, of course, when you looked at that human, compared to the mouse, you would be tempted to argue that's a completely different set of fat pads that the human display compared to the mouse that we use, obviously, as a model organism. Now, actually, it turns out to be not so different when you look with the same methodology. So what we've done here is basically also injected a radioactive glucose, or in this particular case, it's a radioactive lipid, and actually imaged this before and after stimulation. And you can actually see here these areas that light up. The biggest one here is the brown adipose tissue in the back, but you have many other additional signals that pop up. And we're fairly convinced that these are actually fat cells, little mini fat pads, because we know that when you use now a different method in which we genetically eliminate the fat cells, this is a control mouse that you've just seen. This is one without fat. You can actually see that all these dots disappear, and all the lipid actually starts to pop up in the liver, exactly like in a lipodystrophic individual. So based on things like that, we are very excited, and this is very recent data, actually, in collaboration with Oran Haas, and we actually were able to define now a host of new fat pads in the mouse, which are exactly reminiscent of the ones that we see in humans. And the ability to do this in the mouse allows us, of course, the possibility to go in, harvest the tissue, characterize the tissue, and find ways by which we can actually interconvert these individual components. So that's definitely another new aspect of fat tissue that really only popped up both clinically and preclinically in the last couple of years. Another new aspect that I want to point out to you is the ability of the fat cells to undergo additional unexpected changes. So as we differentiate the fat cell from a precursor cell to a mature cell, conventionally we would think we only have a limited number of possibilities what that fat cell can actually do. It can either increase in size, so you overeat, it's going to increase in size. You go on a diet, it's going to shrink in size. And that's really the conventional scenario. Most of the time, these fat cells don't go away. Once they're there to stay. And in our case, in humans, they actually have a half-life of nearly 10 years. So what you carry around today, you probably developed about 10 years ago. So they shrink, they increase in size, and on rare occasions, in fact, they can actually die. But that was pretty much the whole repertoire of what a fat cell can do within your system. However, the last year or so has also revealed that there are additional possibilities that a mature fat cell can actually retrovert to a precursor cell, or even worse, a fibroblast or myofibroblast that I'll mention in a second. So they can go back and forth, and they can actually be recruited again. And that sounds like a relatively trivial finding, but it was rather unexpected, basically, for the field in that sense. So that helped us initially actually define a process that we call AMT. So that would be the adipocyte myofibroblast transition. And this is a little bit of a play on what the cancer people refer to with their EMT, which is another process that cancer cells can undergo and do important changes. So we saw that, for instance, initially in the skin. when you expose the skin to sunlight, UV damage basically, your dermal adipocytes, this is an interesting layer of fat cells that you have in the skin, they can actually undergo this dedifferentiation process and turn into these fibroblast precursor cells, and then these precursor cells can actually turn into what we call myofibroblasts. And whenever you hear myofibroblasts, you have to think fibrosis. So those are the cells that actually cause the deposition of extracellular matrix component in the skin, and they're actually responsible for sclerosis of the skin, which is, again, an important pathological conditions. But at the same time, of course, you can also see this on the more quote-unquote normal physiological conditions. So in this particular case, we have taken the fat of a mouse before pregnancy and genetically painted every single fat cell in blue. Now, the mouse gets pregnant and starts to get the mammary gland ready for lactation. And as you can see here, these milk-producing lobules in pink start to pop up. They're still surrounded by these blue fat cells. And as we go on, actually during lactation, what the textbook would suggest that all these fat cells simply disappear. It turns out they don't just disappear. They pop up as labeled blue fibroblasts now. And when the mouse undergoes involution, which is basically it stops lactating, and the fat is coming back, the lactating lobules disappear, the fat is coming back, and guess what? Every single fat cell afterwards is still blue. That means those are the same fat cells that were there beforehand. And you can repeat that process over several pregnancies, and still after a number of pregnancies, the vast majority of these fat cells remain blue. So that means they have the ability to go back and forth between this pre-differentiated state and the fully differentiated state as part of a perfectly normal physiological response to lactation. But again, it can also turn pathological in the same tissue. Breast cancer. You have a lesion formation in the ducts. They will break through the basal lamina. They will infiltrate now the surrounding adipose tissue. Again, in this particular case, we've labeled the surrounding adipose tissue with a red color, and we have infiltrating tumor cells. And as the tumor progresses, it will actually convert the fat cells into these little dots here, which again, those are fibroblasts, and it turns out those are myofibroblasts. And these myofibroblasts, again, contribute very significantly to the fibrosis that occurs during the progression of the tumor infiltration and growth. So very important contributors to the pathology over the course of breast cancer. So it turns out whether physiological, pathophysiological, or under normal conditions, the fat cell has the ability to actually undergo this dedifferentiation program. We call it the Brownian motion of the white fat cells. It goes back and forth, even under normal conditions, at a certain low frequency within the tissue. And that offers, again, wonderful new ways for intervention and potentially even weight loss under those conditions. Now, let's leave the fat cell per se a little bit and look more systemically. What does the fat cell actually contribute systemically? We mentioned these hormones, these adipokines, some of which we've defined. Others have defined additional ones. And what we've appreciated, probably exactly since about the mid-90s, that the fat cell is an endocrine cell. It's a professional secretory cell that produces a lot of constituents for the system. Some of these constituents act locally. Again, I mentioned the ability to modulate the local extracellular matrix. That fibrosis is very important. There's a lot of pro-inflammatory factors that the fat cell can produce and pour out into the system. And then last but not least, that last category of adipokines, so these marker proteins that are highly selectively produced at the level of adipose tissue and released into the system. And one of our favorite ones here is this protein called adiponectin that we were fortunate to define for the first time quite a few years ago. And it's really been the workhorse for the lab to actually define adipose tissue physiology in very, very many different ways. So it really has taught us a lot. Now, every single one of the factors that fat cells produce will circulate at higher levels the more fat you have, with the exception of adiponectin. So there's an inverse correlation. It's a bit counterintuitive, but there's an inverse correlation between fat mass and adiponectin levels in your system. The more fat you have, the less of it is actually there. And that already tells you that this is probably a pretty good thing to have around. And you can go back to our individuals in the clinic that are fat and fit versus somebody who is simply obese. You can adjust for the exact same body mass index, so same amount of fat. In one case, it's happy fat, in the other one, it's unhappy fat. And what happens here is, of course, that the ones with functional fat will have higher adiponectin levels in circulation. The ones that have dysfunctional fat will have lower adiponectin levels. So what it really tells us when we measure adiponectin in circulation is not so much how much fat we have. It sort of serves as an excellent integrator of the fat tissue health. It takes a snapshot, interviews these fat cells, and if you have high adiponectin in the system, across the board almost, it's a reflection of very metabolically flexible adipose tissue, highly insulin sensitive, and it actually transpires into the entire system. This has really given rise to thousands of papers, tens of thousands of papers basically, of papers that use this as a marker, papers that study the function of adiponectin, and there's almost not a single cell in the system that's not directly or indirectly affected by the actions of adiponectin. I could now spend the next couple of hours talking through the action of adiponectin in every single tissue. Let me just pull out one example because it's a collaborative effort again on campus with Orson Moe's group here in nephrology and internal medicine, the effects of adiponectin on the kidney. So again, if you do this in the mouse, we have ways of providing an insult to the kidney. There's a specific subtype of cell in the kidney, the podocyte, that we can manipulate. And when we manipulate and provide this insult to the kidney, it becomes dysfunctional. You have a lot of protein that these mice pee out. And obviously, ultimately, that will be detrimental. And it is detrimental if the mouse doesn't have any adiponectin in the system. In fact, the kidney becomes, again, very fibrotic under those conditions. But we can avoid that if we have a mouse that has a bit of an excess of adiponectin, and they will actually quite readily recover from this insult and reconstitute normal kidney function. And you can go down now a long list of different tissues, and across the board you will actually find the beneficial effects of adiponectin on the system. So we've learned with this type of experiment, we've actually learned an awful lot about adiponectin action in the different tissues. We appreciate that it's potently protective from cell death, so we call it anti-apoptotic. It can actually generate vasculature, so it's pro-angiogenic, and again, even for fat tissue itself, it's actually highly protective. It is anti-inflammatory, for instance, so again, a beneficial effect, and you've just seen an example of the anti-fibrotic action of adiponectin, very important for the kidney, very important for the lung, very important for the liver, in fact, in that context. It can actually create the generation of more fat cells, and again, that's actually not a bad thing because you, again, prefer to have more fat cells that are smaller than fewer ones that are bigger. And last but not least, with all of that, it actually creates an increased level of insulin sensitivity. So it's not just a marker of insulin sensitivity, it's a driver of insulin sensitivity. Now, the epidemiologists also have fun with it because it really gave us the possibility to use it as a biomarker in the clinic. So it's a very good biomarker for existing metabolic disease. You're type 2 diabetic, you're going to have lower adiponectin levels across the board. And as your insulin sensitivity improves, as you go on an anti-diabetic medication, the adiponectin levels will go up. It's also actually a good prospective marker for the development of diabetes and cardiovascular disease. So even if you are normal today, metabolically speaking, but you have low adiponectin levels, that will actually lead to an increased risk to develop type 2 diabetes in the near future, more or less. Now, how does a single protein do all of these things? And I warn you now, just for 2 minutes, this is going to be a little nerdy lipid chemistry here. The ones that we were prompted to study in this particular context is a special class of lipids that we refer to as the ceramides. Now, some of you might know ceramides because almost every single skin lotion contains ceramides. They're important for the integrity of the skin. We like to rub them in on the surface, but when these ceramides are actually present in other cell types, in other organs, they can actually constitute a big problem if in excess. So you associate excess ceramides with cell death, and that's particularly not good in the case of your cardiomyocytes, your heart cells, the beta cells that produce insulin, and even in the liver. That is a big issue because they can actually cause insulin resistance. They are associated with inflammation. The bottom line there is you do not want elevated ceramides in your tissues. But the good news is there's a single conversion of these ceramides to another class of esoteric lipids called the sphingosines. And they're actually exactly the opposite of what the ceramides are. They actually signal survival, proliferation. So the cancer people are interested in these lipids because they want to lower these and increase these because they want to kill the cancer. But from a metabolic perspective, we want to lower these ceramides and we want to increase the sphingosines, which are beneficial in that sense. And to make a long story short, yes, adiponectin will actually reduce these ceramides through a number of different mechanisms. And that's the key driver for the action of adiponectin in all of these different tissues. A reduction of ceramides in fat cells, or for that matter, in every other cell, will improve the diabetic phenotype. When you reduce the ceramides in the liver, you can actually improve the insulin sensitivity. So with the reduction of the ceramides in these tissues, you can see over here, the lipid-laden livers here convert in the presence of adiponectin to livers that have a lot less lipid stored, and as a result, they actually improve their insulin sensitivity. And without going too much into the nerdy details here, the adiponectin proteins, they have receptors that they interact with on the cell surface of cells, of target cells, of the liver, for instance. And when they engage that receptor, they will actually specifically activate an enzymatic activity, and that enzymatic activity will actually convert these nasty ceramides into the beneficial sphingosines and sphingosine phosphates. So that's definitely something which we want to explore further in future studies as well. Your legitimate question is this of relevance at all clinically? Does the same thing happen in humans? And again, the short answer to that is absolutely. We can actually see the exact same correlations. In this particular case, we can't do cause and effect, but they correlate very well in the sense that elevated levels of ceramides in the system in humans, we can measure them in plasma, we can measure them in biopsies. Elevated levels of certain subspecies of these ceramides are tightly associated with insulin resistance. So there's a whole new axis in which we can think about anti-diabetic approaches by playing with the levels of adiponectin, by playing with the activation of the receptors, lowering the ceramide content of these target tissues, which will be anti-steatotic, so it will drain the liver of the lipids. It will be anti-fibrotic, and it will, last but not least, improve insulin sensitivity. So we've covered here some of the lipids. We've covered some of these hormonal factors. There would be more, and I just want one quick snapshot of another component here, the metabolic intermediates. And again, we don't give the fat cell enough credit for the contribution it makes, particularly in the fasted state, which is really the sole source of a lot of the systemic components that are floating around out there. And again, another nerdy component that we love and to embrace here is uridine, for instance. Uridine, if you open a biochemistry textbook, you will realize this is a basic building block for a lot of different components. Conventionally either absorbed through the nutrients. In the fed state, the liver produces all the uridine that we need. However, in the fasted state, the liver actually stops producing uridine, but the system still needs uridine desperately, actually. In fact, in the fasted state, it needs even more, and it is the fat cell that will start pouring out uridine into the system under those conditions. And the fact that the fat cell will produce these in the fasted state has a whole series of very important implications, including, for instance, it drives that phenotype where we, in the fasted state, actually lower our core body temperature a little bit, and that, at least in the mouse, is the direct impact of having elevated urine in the system. And after a meal, you actually feel a little warmer. It's very slight. It's only a degree and a half, but that's actually a result of lowering uridine levels after a meal. So that's all very important. And when we start thinking about the bigger picture in terms of putting our pharma colleagues' hat on here, what could we really think about of the fat cell as a target for intervention for metabolic disease? You can come up with a very large number of possible areas for intervention that the fat cell offers, and it's certainly one of the key targets at this point to find new anti-diabetic and potentially even anti-obesity medications in this particular context. Now again, I referred to it a couple of times because I feel a little defensive being a mouse doctor up here, sort of lecturing clinicians about the use of the mouse and how valuable it is. And it's always an important question, is any of the things that we do at the preclinical levels, does that really translate into something clinically useful? And we need both, basically, of course. What we can do now at the preclinical levels is we have the ability nowadays to actually interfere very specifically. We can make genetic modifications in a specific cell. And not only can we do this, we can actually also time that intervention very well. We can turn things on and off. And that, of course, allows us to ask questions related to cause and effect. And what we love to do in the lab is basically intervene genetically, bring the system out of balance, and actually watch it defend itself as it reaches a new homeostatic equilibrium amongst all of that. And that can be extremely educational. And I want to just give you at the end now one last little example where we can actually illustrate this very specifically. And I picked an interesting case because it's going to be something that you're familiar with, amyloid precursor protein. We all have heard about Alzheimer's disease. This is thought to be one of the causative proteins of Alzheimer's disease because it forms these plaques in the brain. And when you hear APP, amyloid precursor protein, you're thinking Alzheimer's disease. But is this really only relevant for the brain? Because as it turns out, it's actually expressed in many other tissues as well, including, and this is an old paper from almost 10 years ago, that reports that in an obese fat pad, in obese individuals, they start to overproduce APP. Now, this could be a little side observation. Should we care about this? Should we not care about this? I think we should care about this because it turns out when you take this observation back into the mouse and look at that in the mouse, you will actually see, when you compare now, this is a chow-fed lean mouse here, this is a high-fat diet mouse, and you look here for the levels of this protein, you can actually see that the obese mouse, just like an obese human being, will start to overproduce APP. But what's curious about it, it's not necessarily secreted. It's meant to be released from the cell. It actually ends up in the mitochondria of these cells. Now again, should we care? Well, we can certainly prove by any number of methods that this is indeed a mitochondrial protein. This is a control here. Here you can actually see how APP integrates into these little power plants of the cell that we also desperately need to produce energy. And that APP molecule actually ends up in these mitochondria as seen here. And when you look at this a little bit more carefully, you can actually realize that it's basically just like a really obese Santa Claus coming down the chimney. This protein gets stuck on the way in. There's an import channel. It gets stuck and blocks this import channel. Now, the mitochondria have many, many import channels, but at some stage, you're going to saturate every single one of these import channels, and you're going to create a dysfunctional mitochondrion. And that is not only the mechanism by which this is really bad news for the fat cell, it could also actually be the mechanism by which our neurons suffer, not so much from the deposition of these plaques outside the neurons in the brain, but much rather it could actually be a function of getting this stuff clogged in the import channel. Now, if I go way, way back in my time as a Ph.D. student, we actually used something similar at the time. We got things stuck in the mitochondrial import machinery, then cross-linked whatever is surrounding it, and we actually used that methodology to pull out the surrounding channel proteins for the mitochondrial protein import. Now, this was completely artificial. This is now a lot more physiology associated with it. And again, in the mouse, you can give and take at will. So when we give a little bit too much of this protein, just like what you see in the obese state, you can actually see that as soon as we turn this thing on, the mouse starts to gain a lot more weight. And with that increase in weight, it deteriorates metabolically. Not only that, you can see as you go from the baseline to 7 days of treatment with more of this protein, to 14 days of treatment, you can actually appreciate how the small fat cells turn into really, really big fat cells. And they're beautiful to look at, but they're totally dysfunctional. So you don't want that, and we believe that this is actually something that's going on in the obese individual, in the periphery, in the fat, and we conclude from that that the ability to burn lipids in your mitochondria to generate energy is absolutely critical for the health of the fat cell. And if you don't do that, your fat cells are going to increase in size and they're going to deteriorate in their function. So there's actually a very simple bottom line here. Many people argue that Alzheimer's is the diabetes of the brain, and that's true. Alzheimer's may very well have a strong metabolic component to it. There's a lot of data to support that. But at the same time, I dare to propose that diabetes may be the Alzheimer of the fat cell, basically. So it produces APP as well, and in the process, it becomes very, very dysfunctional. So I just picked a few examples out of the hat here that we can talk about more if you want in the Q&A section of this. But I hope I've given you just a little bit of an overview for the importance of the fat cell. The fat cell is our friend. We need it. We need to have it functional, because if we don't have it, all these excess lipids that we consume on a constant basis are going to end up somewhere else. So the role of the fat cell in our metabolic health is undisputed. And it will interact through these hormones, these adipokines, through the lipids, the ceramides, as well as the metabolites, the uridine, for instance. It will interact with a lot of the critical partner tissues within the system and help them maintain normal health. But there is still so much more that we need to learn. Again, you go back to the clinic and you look at some of the lesser known, but very prevalent, quote-unquote, fat diseases. I want to remind you of something that we've actually gotten very much interested in, lipedema. I bet you many of you have never heard of lipedema, but depending on whom you opt to believe, anywhere from 0.5 to 10% of all women actually suffer from some form of lipedema, which has no clear-cut defined etiology. We don't know what causes the disease. It's frequently associated with obesity to some extent, frequently misdiagnosed as obesity. But this fat here that you can see with the characteristically ending right above the ankles here, and eventually it starts out only in the lower extremities, eventually it will be on the upper extremities. It is very curious that, for instance, when these women go on a diet, this fat does not disappear. There's no lipolysis going on. So the only way you can remove this is through liposuction. And it's obviously something which is horrible for the individual to go to. And we need to better understand this disease phenomenon. And that's now where we work closely together and are actually supported by funds from the Lipedema Foundation to further define the pathology of this adipose tissue. I also want to remind you of the role of fat in the context of infectious disease. So there are a variety of parasites that we are exposed to. One of which is a parasite called Trypanosoma cruzi. And some of you may have heard of Chagas disease, which is a heart disease. It's prevalent in Central America and in South America. It's caused by a little beetle called the kissing bug. And it's going to transmit these T. cruzi. You're going to have a severe fever as you're being infected. Chances are you will survive it, and then it subsides. But for the rest of your life, you're going to have these T. cruzi parasites inside of you. And whenever you might be immunocompromised or for some other reason, you have another outbreak of these decades, potentially decades later. And in the process of carrying these parasites around for decades, you actually develop a very severe form of heart disease. Now, since you develop heart disease, it was thought that the primary residence of these parasites is actually the heart cell. But it actually turns out that the predominant reservoir for these parasites is the fat cell. And there is no better place to hide away for decades than a fat cell, because you're protected from the immune system. This is a cell that hangs around for decades, if need be, and you basically are in a candy store because you're right next to a lipid droplet. So understanding this disease is very important. Again, you would argue, why should we care? Well, depending on your position, or actually independent of your position on global warming, the fact is this disease is migrating north. We have cases of primary infections in Texas now, so we should care about Chagas disease, and in that sense actually understand the basic physiology of this parasite behind it a little better. And unfortunately, I did not have time to discuss this further, but there is this interesting correlation between increased body mass index and increased tumor incidence, almost across the board. It affects almost every single cancer, some of them very much prone and driven by obesity. Endometrial cancer, breast cancer, postmenopausal breast cancer, colon cancer, pancreatic cancer, all emerge as with an increased rate when you're obese, and your prognosis in the obese state to get cured from that cancer deteriorates. What is the relationship between the fat cell and the tumor cells? And again, it's particularly striking in the context of breast cancer, where these tumor cells go through the basal lamina, are in the stroma, and in direct physical contact with the tumor cells. So there's a crosstalk. We are trying to figure out, with our colleagues in the Cancer Center, what is the interaction between the fat cell and the tumor cell? Does a pathological fat cell contribute to that interaction, and why does it predispose us to this increased cancer incidence? So all of that are still questions that we don't have good answers for, but what we do know is that we want to keep these fat cells happy. It's very simple. Happy fat cells, the fat cell is happy, everybody's happy in the entire system. Once they get inflamed and angry, they can actually inflict a lot of damage onto the system. So with that, I want to really acknowledge the work of many, many in the lab over the years. I want to flash up the names of the individuals that are currently in the lab and contribute to what I've just shown you. I want to flash up a few more recent graduates that moved on, and then this set of postdocs here, all of which have gone off and started their own labs here and elsewhere in the world. I want to acknowledge two of my major partners in crime here, Joel Elmquist and Jay Horton. We run sort of the local sort of metabolism program. Two additional people that I want to acknowledge, Perry Bickle and Scott Grundy, very important programmatically as well. The faculty in the Touchstone Center, which I'm really very proud of, an exquisite group of people, which is highly interactive, and we really cross-fertilize each other, and most of which are actually in the audience here. And all of us here are supported by an exquisite group of support group that helps us with a smooth operation. And I can't give enough credit to the intellectual climate here on campus with important collaborators. If you're in the audience and you're not in the acknowledgement slides, I really apologize, but there's a large number of people within internal medicine and on North Campus elsewhere that with whom it's a pleasure to collaborate, as well as other key collaborators in other institutions. And last but not least, as Dr. Podolsky mentioned, I want to acknowledge the people that support us, the Touchstone family, the O'Donnell Foundation, and of course our grants agency, the NIDDK, the ADA, the American Diabetes Association, the JDRF, and last but not least, also CPRIT here in Texas that sponsors some of our cancer work. With that, thank you very much for hanging around and bringing it.
And thank you for leaving some time for some questions. I'd like to open it up to over here, please. Yes.
So if the dividend is overexpressed and that leads to more glucose metabolism, more fat metabolism, what do you know about or what can you do to prevent like ketoacidosis?
To prevent what?
Ketoacidosis or like ketosis, entering ketosis?
Right, so you
Phil,
improve the
Phil,
insulin sensitivity.
Phil, why don't you repeat the question because people behind me.
Absolutely. So if adiponectin stimulates glucose metabolism and fatty acid metabolism, how does it avoid a phenomenon that we see in type 1 diabetics that drive most of their metabolism off fatty acids because of the lipolytic event, referring to as ketoacidosis? So again, the emphasis is that we are making the system generally more insulin sensitive. So as such, I would argue we don't preferentially actually favor the burning of lipids only, but it also refers to the burning increased TCA cycle glucose sugar metabolism as well. So it is not something that we see. In fact, adiponectin is actually protective against ketoacidosis. If you use excess adiponectin in a type 1 diabetic mouse model, it is potently protected from the ketoacidosis. So even in a type 1 diabetes setting where you have limited amounts of insulin available, that's when you really unmask the absolutely essential nature of adiponectin.
Questions? Phil, the evolutionary origins of brown fat, does that associate with the development of warm-blooded mammals? Is it more ancient than that?
It actually is a phenomenon that you see mostly in I guess you refer to euthermic animals in terms that the ones that have to keep warm. So you see it predominantly in that. They actually have a distinct precursor source for these cells. So they are not necessarily derived from the same precursors that our white adipocytes emerge from. They are separate. And obviously these tissues play a role under normal conditions, these brown tissues. They play a role during hibernation as well when you're doing the awakening. So you can turn this on and off. And it's an amazing capacity to heat up the system and turn it off again once you have these brown fat pads localized. That's why it's such an attractive perspective to tap into that and perhaps just burn energy in a futile cycle without the necessity to move. It's a bit of an illusion perhaps, but it's certainly an attractive thing to think about.
I think there was a question back there, yes.
Can you talk about how fat cells respond differently to either exercise or dieting?
The conventional wisdom in the field is that it's sort of a dogma. Fat reduction is always symmetrical. So the idea that you can get rid of your belly fat by doing a lot of sit-ups is probably an illusion. You should still do sit-ups, but you're going to lose that fat symmetrically across the board. The same holds true for exercise. Exercise is also, and I hate to disappoint all of you, exercise is not necessarily a regimen to lose weight. You should exercise for many reasons, but not necessarily to lose weight. You want to lose weight, you have to eat less. It's really the caloric intake that matters there. The same thing, and it's a good point that you raise, I want to clarify when I talk about these very obese mice, or I talk about these fat but fit individuals, I don't want to have this misconstrued as an endorsement for obesity. you still don't want to be obese, but you can get lucky. And you have to view the fat cells, again, as a cell type that comes to assistance during excess caloric intake. And it takes the hit for the system by storing the excess lipids and protecting everyone else from having to store these excess lipids. That's why I say the fat cell is our friend. But we keep providing insult after insult after insult, and eventually these fat cells just say, okay, I can't take it anymore, and they'll burst and they become inflamed, and they do all these nasty things. But they are trying to protect the system.
Well, I think with that, we'll thank Dr. Scherer. I would invite everybody to visit with him on the McDermott Food Court on the first level here. And also at the same time, I would encourage you to put on your calendar April 26th, when we'll have the next President's Lecture. Our colleague, Dr. Biff Palmer, will talk about his experience climbing the highest peak on each of the 7 continents and what he learned about physiology in the process. So we'll look forward to seeing you on the 26th, and just now to join Dr. Scherer on the first level. Thank you again, Phil.
Thank you.